Great.
Okay, so what I'm going to talk about today is the human microbiome and its interaction with different mucosal surfaces. So next slide, please. Okay, can we have the whole slide up? All right, that's it. Okay, so I'm going to talk today about 4 different mucosal surfaces. Of course, there are many more in the human body, but these are ones that I've chosen to give an example. The point at the bottom that you'll see is that all of these 4 mucosal surfaces show an abnormal microflora in disease, and I'll check that as well as we go along. So next slide, please. Okay, Vlad, next slide, please.
Okay. Is that the next slide?
Yeah, that's fine.
Right.
Okay. So what we're going to talk about is the mucus barrier. So this is essentially a surface and adherent mucus gel. It's a dynamic, renewable protective barrier which protects against the external environment. And it's also an integrated defense system, which includes a biofilm and protective proteins as well. Now, I'm not going to talk about the protective proteins. So it's a surface gel barrier which interacts with the microflora that is present at each individual mucosal surface. So next slide, please, Lad. Good. Okay. So here's a picture just to show you what it is that we're talking about. If you look at the bottom, you can see the brown lump is a cell, and the square at the top represents the secreted mucus gel layer. Now, there's between these two, we've got on the surface of the intestinal cells, we've got membrane-associated mucins. They link up with the gel layer on top, which contains the secreted mucins. And these mucins, they have a number of different properties. They bind water, so they have hygroscopic effects. They can filter. They act as a network. They filter anything that comes onto the surface of the gel layer, and they'll filter those things out. They have charge, positive due to protein, negative due to the sialic acids and sulfates, and they have stoichiometric power. That means that the glycans that are expressed on the proteins that are present have a range of different glycan structures, and these are repeated quite often. So there are a number of different glycan structures which are available for binding of all sorts of components that are in that gel layer. So next slide, please, Lad. Can we have the next one, please, Lad?
I don't know why there's a little bit of delay. Can you see the slides called Bacterial Flora and Mucosal Surface?
That's good. That's okay. Fine. Right. That's what I need. Okay. So the bacterial flora regulates the expression of particular carbohydrate structures, glycotopes, on the surface barrier. And this is part of the relationship between the symbiotic and commensal bacterial species in man. This has been termed the glycan legislation, and this is Jeffrey Gordon in the States proposed this for the human gastrointestinal tract, but of course it's relevant for other mucosal surfaces as well. So next one, please, Vlad. Yeah, it's a bit slow. Okay, so this is the relationship that we're talking about. The relationship of the host with bacteria, whether those are symbiotic, commensal, or pathogenic. And the common link is the mutualism between the commensal and the pathogenic. And these are the driving forces in coevolution for the development of a bacteria-host relationship, because we have bacterial genetic diversity, we have a capacity for rapid growth of the bacteria, and we have a high population density. So next one, please, Vlad. Okay, that's good. So in order to evade pathogenic microbes and to create a symbiotic relationship with the indigenous bacteria, there's a strong selective pressure to create a diversity of glycan structures, and this is where the evolution aspect comes in. So next, please. Okay, so here's a picture to show you what's going on. So at the top we see the host mucosal barrier. The purple layer at the top is the lamina propria, beneath the layer of intestinal cells, the epithelial cell layers, and in green you can see the secreted mucus. Now, if we look at this in more detail, down at the bottom on the left-hand side, you can see that the secreted glycoprotein can be degraded by glycosidases, which are being secreted by the bacteria. The bacteria is the yellow square at the bottom right-hand corner. Now, part of that those monosaccharides, which are digested by the glycosidases, will be picked up through the permeases and taken up by the bacteria. They then use some of that for energy production, but it can also switch on the genes which produce and secrete the glycosidases. If the level of carbohydrate, the monosaccharides, gets too high, then it switches off. So there's a repressor built into the system as well. So it's a neat cycle to maintain and control the expression of glycosidases, mucus degradation through the bacterial system. Okay, next one, please.
Can you see the slides?
Okay, yes, the next one's come up. Okay, so this is a micrograph again to illustrate the mucus, the mucosa. At the top, you can see the mucus gel barrier. You can see the thin blue line of the glycocalyx, and you can see the goblets, the goblet cells all stained up with the goblet cells, the mucus vesicles in the goblet cells, and the lamina propria below. So next one, please. Okay, good. So we know that the human large intestine contains mucus glycoproteins rich in sialic acids, and we can detect those readily using the mild chlorhydric acid Schiff histological stain. And the next couple of slides are to show this this feature. So next one, please. Okay, that's good. If we do a direct stain using the pyridic acid stain, we don't stain up all of the goblet cells. The reason for that is that the sialic acids in the glycoproteins, the mucus glycoproteins, are acetylated on their 7 to 9 tail. Now, in order to detect all of the sialic acids, we do a mild saponification, about 30 minutes, 0.1 molar sodium hydroxide, and the next slide will show you the what happens if we do that. There we are. That's good. That's then you can this stains up all of the goblet cells, the sialic acid in the goblet cells in the whole of the mucosa. So we can see that quite nicely. So next slide, please. Okay. So yeah, I think right. There's a bit. That's right. Okay. So in order to be able to study this in more detail, what we linked up with was the people in the surgical department. They do an operation where, as part of the therapy for inflammatory bowel disease, they isolate the a part of the intestine and leave it with no contact to the fecal flow, so that they have a bag and the feces comes out separately. And this is maintained for 3 months. So it means that there is a section of the intestine which is still in the patient, which is completely devoid of contact with the fecal microflora for all of that 3-month period. So this gives us an opportunity to assess mucosal glycosylation with and without contact to the microflora that's present. Okay, next please. Brilliant. Okay, so what we've got here is that we screened through a whole range of different carbohydrate structures using either lectins or antibodies. And here, for example, you can see for Silar TN. On the left, you can see where there was contact with the with fecal flow, the normal situation, non-diverted. And we can see, as we would expect, the Silar TN is not expressed in the normal mucosa. However,
sorry, Tony, what's TN?
It's just a nomenclature for the it's T antigen, TN antigen, and it's this disaccharide structure that you can see underneath it. It's just a name that has been used. Okay, so okay. On the right hand side, you can see that if we look at the diverted, where there is no fecal flow, we see that there's an upregulation. This particular structure, Silar TN, is expressed. If we go to the next one, please. Here, this is a a structure which you can see at the bottom with a Galnac structure called SDA. This can be detected with this lectin, and on the left you can see normally that is expressed. So we see a strong positive. If we look in the diverted with no fecal flow on the right-hand side, there's a downregulation. It's lost. And the next slide. Brilliant. This is the same structure again, the SDA antigen, but this time using an antibody. So confirmation of the first result with the lectin, we see the same result. There's a down regulation. So next slide, please. And what we did is that we used a number of different reagents to screen for different carbohydrate structures. The carbohydrate structures you can see listed on the left-hand column, with the and the antibodies, the reagents in the second column, antibodies at the top, lectins at the bottom. All of these would normally give positive results as our control. In the diversion, in the final column on the right-hand side, you can see there is a selective response. Some of them change, some of them increase, some of them decrease, others don't change at all. Now, if we go to the next slide, here we can see if we look at the mucin peptide structures using, again, a series of different antibodies, where the controls are as normal, and in the diversion there is no change at all. So the result here suggests that there is a genetic effect. If we look at the genes that control the glycan structures, these are affected in part by the microflora, but not the mucin protein peptide changes, epitopes. So next slide, please. So, in other words, what this means is that the Gordon hypothesis does apply certainly to the gastrointestinal tract, and gives us the link to the genetics in response to the microflora. Next slide, please. Okay, ethnicity also is significant. We carried out a study looking at South Asian and European patients that had inflammatory bowel disease. And there are differences in the expression of the colonic mucins in these two groups of people. So in other words, it does depend on ethnicity, the type of response that you get to the microflora. And the other thing that came out of this study was that European patients with IBD have a higher risk for colorectal cancer than the South Asians. Okay, next slide, please. Okay, so if we move on to the Oculus Surface now, again, just a brief overview. Here, this is the structure of the Oculus Surface. At the bottom, you can see the epithelium. You can see on top of that the glycocalyx, where the membrane-associated mucins are anchored into the surface epithelial cells. And on top, you can see an aqueous layer, which is coated by meibomian lipids, which prevents evaporation of water from the top. But effectively, we've got a mucus layer, a gel layer again, which is present in the eye. Next slide, please. Okay, right. This is a slightly higher magnification, just to illustrate the point. You can see the big goblet cell right in the middle, and the purple blotches there, they represent the secreted mucins which are coming out of the cell into the aqueous layer, sitting on top. And you can see at both sides the glycocalyx with the membrane-associated mucins there as well. So next slide, please. Okay. This is a representation of interaction of influenza virus with the ocular mucins. Just to show that if you look at the cells right in the middle there, if you move from the left to the right, you can see that the cells are invaded by the virus to give the pathology. So next slide, please. Okay, so here we have a structure-function relationship which affects mucosal pathology. We looked at different species, and dogs, rabbits, and humans, and we found the same pattern. So the secreted mucins are an individual example with many targets on the ocular glycobiology that remain to be worked out. So next, please. Okay, so here is effectively what happens with oculomusins. The O-glycans that in the oculomusins are predominantly sialylated, with 85% of the glycan structures, core 1, core 2, are having sialic acids. Now, this links nicely with the known pathogens in the eye, which show links to the sialic acids either through 2,6 or 2,3. The 2,6 links are targets for Pseudomonas aeruginosa binding, which is well known as a pathogen on the ocular surface. And in addition, the 2,3 linked sialic acids are receptors for several different viruses, including influenza virus. Next slide, please. Okay, so if we now move to the female reproductive tract, we carry out a number of studies with this. We looked at the normal vaginal and cervical mucus barrier, and we looked at the microflora that is present. Under normal situations, the majority of the microflora are lactobacillus strains. It has the female reproductive tract has a characteristically low pH, down at pH 4. And that is the normal pH which is maintained there. And what we know is that there are glycosidase activities that are secreted by the lactobacillus strains, which are sufficient to carry out normal turnover and degradation of the cervical mucus. So next slide, please. In the disease state of bacterial vaginosis, which is a common infection in adult women, we find that the women with this infection have an increased pH at their mucosal surface, which is around 6 to 7. What happens as a result is that the microflora changes. These women have a loss of lactobacillus species and have an increase in anaerobes such as Gardnerella. As a result, these anaerobes, they have higher levels of glycosidase activity, and it leads to the destruction of the mucus barrier in the vagina and the cervix, and you lose protection and interactions that would normally take place, including fertilization at these locations. Okay, next slide. Next slide, please.
Yeah, is it there?
Not, yeah, there we go. There we go. That's it. Okay. So this is a list of the strains that you find in bacterial vaginosis. On the left-hand side, you can see Gardnerella at the top and a number of related strains that are found. Most of them are gram negative. And if you look over on the right-hand side, you can see that these major strains, they all contain sialidase activity, sialidase positive. And the ones with the question mark, I think, are now have now been shown to contain sialidase as well. So next slide, please. Can we get the next one?
It's on the next one for me.
Okay, that's it. That's it. So the clinical background to this is that the routine method that is used in clinics is to use the gram stain. The gram stain is a chemical stain. What happens is that you take a swab, you smear it onto a glass slide, and then you stain it with the gram staining process, and this gives you a color. These are then graded 1, 2, and 3. 1 is effectively normal, and 3 is strongly bacterial vaginosis positive. However, this is difficult because you have to prepare the slide, you have to stain the slide, you have to get someone to look at it to decide whether it's positive or negative. So people that are coming into the clinic to be tested whether they got bacterial vaginosis are not going to come back for a second visit, and they're not going to hang around for ages. So we thought that we should develop a rapid assay using sialidase, which would make this a little more effective and certainly more practical for the clinics. Okay, next slide, please. Okay, so here's the bacterial vaginosis stain. On the left-hand side, you can see the grade 1, and up at the top, you can see a whole load of lactobacillus bacteria that are stained up there. If you go over to the right, those lactobacillus have disappeared, and there are far fewer lactobacillus and only a few other different strains there present in the grade 3 one. So next one, please. Okay, now if we want to screen for sialidase to detect bacterial vaginosis, we have a number of different options of how we can do this. We can use physiological substrates, and these are listed here. So we could use a sialolactose, a trisaccharide. We could use polysialic acid, columinic acid. That has a different linkage, 2,8 linkage rather than 2,3 or 2,6. Or we could use glycoproteins, and these would be the easiest to use in a rapid assay. We could use phetuin, alpha-1 acid glycoprotein, or salivary mucin, or we could use glycolipids, the gangliosides. The ones in red are the two that we chose to use, and these are glycoproteins that have N and O linked sugars. So next, please. Next slide, please. Ah, okay.
Are these all coloured labels, Tony?
Yes. This is the, okay, fine. These are the synthetic substrates. As you can see, the sialic acid in red on the right-hand side, the alpha glycosidic link through carbon 2, and the blue shows the synthetic aglycones, nitrophenol, methyl umbelliferol, and the one that we chose, the bromochloroindolol. These are ones that are readily available, easy to synthesize, and easy to use in a test. So next slide, please. Okay, now the point that we need to make here is synthetic substrates could provide us, or will provide us, with a more sensitive screening option. However, some sialidases have been found not to react with certain of the synthetic substrates that have been prepared. So each time we come up with a new activity that we want to analyze, we should go for both the physiological and the synthetic substrates. So that's the idea that that we adopted, and that's what we actually did. So next slide. What we used for routine detection is this bromochloroindolol sialic acid, and we use that to develop a semi-quantitative test for sialidase in BV patients. So next one, please. Okay, so here's a 96 well plate showing the sialidase activity down at the bottom. You can see the BCIN comes up blue. You can see that. This plate also at the top has got an assay for sulfatase at the top in yellow, but the sialidase activity is the one that you can see down at the bottom. Okay, next. So what we did was to check out how does the spot test correlate with the gram stain. So we looked at 99 people who were either BV positive or negative. That's right, on the left-hand side there. And then we divided up into positive or negative spot tests. And the analysis that came back there showed that in fact that there was a preference for the spot test over the gram stain. Next slide, please. Can we get the next one? Okay, there we are. So here are some figures to go for. These are for the two glycoproteins that I mentioned. On the left-hand side, you can see the grade 1, 2, 3. 1 is effectively normal, 3 is strong BV positive. And you can see there is a strong correlation. If you go to the compare grade 3 with 1 with both of these substrates, there is a significant increase in sialidase activity. So next slide, please. So the quantitative levels of sialidase activity are significantly higher in patients with B. And we found that using the BCIN synthetic substrate was in fact good for screening. But we need to remember that that was the selection and comparison with the physiological substrates which we did before as well. Okay, so and last to come on to the respiratory epithelium, this again is a special is a mucosal surface that has a thick mucus blanket on the top, and is a dynamic system, as all of these mucosa are. The things that I like to point out here are that there are different sites on the respiratory epithelium that have different types of responses. You can see the majority of the cells are ciliated cells. You can see down at the right-hand side, you can see the mucosa with the cilia. Those ciliated cells have contact with the secreted mucus gel, which is at the top of the right at the top of the diagram there, and they beat into that mucus gel to drive it forward and to remove it from the surface. At the same time, we have mucins that are secreted from the glands. If you look in the middle at the bottom, you can see the mucus glands, and the mucins that are produced in these glands are secreted up the duct right in the middle to join up with the mucus gel, which is on the surface. So if we go to the next slide, here, this is a diagram to show you in detail what happens. We can see in the middle with in red, we can see a MUC5 mucus bundle. This is the secretion from the mucus cells in the glands, which produces a large MUC5AB strand or bundle, which is secreted out onto the surface of the respiratory epithelium. At the same time, also in the surface epithelium, though these cells produce MUC5AC, which is a smaller mucin, and this MUC5AC forms a complex round the MUC5B strands, and this is what gives the quality, the properties of the mucus gel, in order for it to be transported by the ciliated cells, which you can see over on the right hand side. So this is the process that takes place in the lung in order to get something which is resistant to the microflora on the surface and to protect it against the microflora in the respiratory epithelium. So next slide, please. Yeah, and here is a confocal microscopy picture, very nicely showing the mucin bundles that are being secreted from the ducts, and this shows very clearly how the bundles are actually formed. Okay, so next slide, please. So, and finally, to go on to smoking, as you would expect, smoking generates a host of different chemical chemical molecules that interact with a whole range of different intermediates on the surface of the lung. The point that I want to pick up is at the bottom. If you look on the left-hand side at the bottom, you can see the ciliated cells and one goblet cell. What happens after exposure to smoke from smoking is that we get a process called hyperplasia. This means that the cells when they are exposed to the smoke, they change and become predominantly goblet cells. This process is called hyperplasia. So the number of ciliated cells is greatly reduced. The number of goblet cells is increased, and as a result, you get much more mucus produced and secreted, which is characteristic for the lung disease. So next slide, that's just summarize that. So hyperplasia leads to increased goblet cell density and mucus hypersecretion, which is the characteristic for lung disease as a result of smoking. So next slide. And that is all of the mucosal surfaces that I'm going to talk about today. I'd like to emphasize again, these are only 4 examples from many different mucosal surfaces that we have through the body. Okay, that's it. Thank you very much, Vlad, and everyone else for being patient.
Thanks, Tony. Thanks, Timo. Timo's got a shoot.
Okay.
Thanks, Tony.
One final comment is I can produce a literature list if people are interested. I can send that through to Vlad. I didn't talk about diets today because it's a bit too much. So that'll have to be a different time.
Part 2. And that was brilliant laser pointer work, Vlad. Again,
yeah, very good.
I'm sorry, there was a delay. I don't know what to do about it. I noticed that when I clicked on the sound, so you can hear me, then it worked. But other than that, I'm sorry.
No, that's okay, Vlad. We got there. I think the message came across. The other thing is I can send you the you've got the PowerPoint anyway. So if people want to have that I'm quite happy with that.
Okay. I've also recorded it, Tony, for Mary.
Oh, okay. Brilliant. Okay.
But, I had the same problem with when Roy Jeffers gave his talk to the MRes students. I had to use this operator slides for him, and he guessed where to put the pointer.
Yeah,
that was quite fun. But it worked.
Yeah. We have to grapple with the technology every day, I suppose. But it's we can manage. We're managing okay.
A fabulous review, Tony. A fabulous review.
Thank you.
And it was like a tour of all the bacterial infections of the different and viral infections of the different, different mucosal surfaces. And that didn't work, like the vaginal one, lactobacillus, and then the ocular, the Pseudomonas aeruginosa, and then you've got the viruses coming in as well. It's each surface was specific for different bacteria.
Yeah, we were fortunate because we had very good links with the Department of Surgery, and I managed to get 2 of the surgeons to do a project in the lab. So that worked out extremely well. Okay, they went back to surgery afterwards, but the links were there, and they understand what it means to get samples that you use in the lab, and that makes a huge difference. If you're trying to squeeze samples from clinicians, it takes a lot of time to explain how.
As long as you keep medics away from doing dilutions, Tony, they're all right.
Okay, that was part of it.
Oh dear.
Okay.
Yeah. Who's Gordon, by the way? You mentioned the Gordon.
Jeffrey Gordon. He's I would say a senior scientist who's I think he works in Washington in the States, but he was one of the first people who actually set up and checked out all of the science tied up with this glycan legislation idea.
This is in the 1990s.
Earlier, I think it was the 80s. I'll send the reference some references through as a backup, and you can check those out.
Yeah. Did he give his name to the Gordon conferences?
No, that's a different Gordon.
Right.
May have been Gordon Bennett, I'm not sure, but maybe that's pushing it.
Gordon Roberts, you know, down Leicester, or Gordon Banks, former England goalkeeper.
Yeah. There's plenty of Gordons around.
Yeah. Sorry, Vlad, I interrupted.
No, that's fine. Do we have any questions? Does anyone have any questions? Jacob, do you have any questions? You said something in the chat earlier.
Yeah, thank you, Tony. That was a fantastic talk. I found in the oral biofilms that domination of lactobacillus drops the pH and forms a carous biofilm.
Yes.
It's interesting that domination of certain bacteria can influence the properties. Do you see an influence on the mucosa as well? So changes in oxygen levels or changes in kind of pH as you go from the top of the mucosal film to the bottom. Do you see changes there?
Yeah, there are changes, but they're much more subtle, and they're managed through the secretions from the epithelial cells. So this is one of the things that in the reproductive tract, in the female reproductive tract, that the production of small chain fatty acids is what mediates the pH. Acetic, lactic acids, and I think that is one of the processes that that takes place. The other side of that is that there is bicarbonate secretion as well, which mediates pH again at most mucosal surfaces. And again, there's a lot of literature on that as well. So in actual fact, pH is quite well regulated at mucosal surfaces.
Oh, fantastic. That's great. Yeah, so I'm currently working with a company called Bac3 Gel, and they're going to send us over some. I've just confirmed a non-disclosure agreement with them, developing collaboration with them. It'd be great to form biofilms on this and maybe see the dominance.
Yeah.
I'd like to get you involved in that work actually, which would be quite good. But,
you're very welcome. I'm here anytime you want.
Yeah.
Just drop an email and I'll
You need to get those density gradient things sorted out first, though, Jacob.
Yes, no, absolutely. But when you get your second vaccine, please do come in the lab. And these are actual gels as well, so it'll be very interesting to see biofilms growing on these. This is fantastic. But thank you, Tony, again. And I'm going to steal your talk and use it.
You go ahead, please do. You're very welcome.
Cheers, thank you, and thanks again.
And Tony, you can come in. It's beautiful here now. You can travel.
I'm waiting for my second jab.
Yeah. Mine's in May. When's yours? Yours must be next month, April.
It should be. Yeah, the second. I should get my second one. That's right. In the next month. Clarice has got a date for hers. She's getting hers on Saturday. Her second one on Saturday.
Because Clarice is 80, is that right? Is she over 80, Clarice?
Yes.
Yeah.
Yeah.
That's right. That's good. My mum and dad have had theirs already.
Good.
Fantastic.
Yeah. So with the SILA, the links, it's amazing that just the change from a 2.3 to 2.6 link changes everything in terms of the recognition. The recognition is that sensitive.
It is, and I think the relevant thing there is that if you just state it as a 2.3 or a 2.6, it sounds fairly simple, and the subtlety is in the protein that carries the glycan with the 2.3 or 2.6 on the end. That is what you have to look at in more detail. I didn't really talk about that. But if you look at the different glycoproteins that are present at the location that you're interested in, then you can see that the 2,3, 2,6 actually comes into play and is very significant.
Mm. Nature is wonderful and a pain sometimes as well. Yeah.
Sorry, I have a question, Tony.
Okay.
Thank you for your talk. It was really interesting. I studied biomed undergrad, so all this sort of stuff's coming back to me, and it's quite good to see it once again. I have more question on to do with a certain bacteria present in Crohn's disease patients and how that would influence the mucosal surface than others, because I'm thinking some could lead to more, if it changes say pH and things, that could have more an effect on the mucosal layer, which could then make it more it could be more compromised to affect something like autophagy, because autophagy is one of these things that then affect the mucosal barrier. And if you affect autophagy through things like carbohydrates produced through bacteria, is this a sort of, like, I know that's a big question, but is it a sort of vicious circle of these kinds of things in Crohn's?
Yes, that's absolutely right. If there are a number of studies that have been done looking at the bacterial populations that you find in Crohn's patients, and what you're saying is exactly right. It does affect the mucosa in a big way, and you can find different types of bacteria that appear to be specific to the IBD Crohn's situation. So the selectivity comes into play again, and I think that is very relevant.
Yeah. Okay. Thank you.
Okay.
I have another question, Tony. When you say bacterial flora regulate the expression of selective glycotopes on the mucosal surface barrier, I'm just trying to get my head around that. So is the bacteria using it as a food source, and then that's that's down regulating it, or is that an up regulation?
It's the bacteria that have been studied have been shown to use the carbohydrate in a form of a cycle. They produce glycosidases and secrete glycosidases that degrade anything like the mucus, whatever it is, in the location that they are. They then generate the monosaccharides. They have permeases that take up those monosaccharides. When those monosaccharides get into the bacterial cell, they can do two things. They can either be used simply as a source for energy production, or and again, this is bacterial strain specific. You find that some of them have a set of genes that are regulated through the presence of the glucose that gets, or the monosaccharide that gets into the bacterial cell, and they regulate whether the glycosidases are produced, synthesized, and secreted.
Right. Yeah.
Okay.
Yep.
And that works as a cycle. If they have loads and loads of glucose, so there's no problem for energy shortage or anything along those lines, then they just switch off. They don't need to produce the glycosidase to create more, so they just switch it off.
So do you think that so a diabetic patient would have a shift in this process?
It's possible. I don't know. I haven't looked to see, but that's worth checking out. Yes.
Yeah, that's quite interesting. No, thank you, Tony. It's great.
Okay.
Okay, Vlad.
Yeah. So the salidases are a good thing to have, right? To have produced by the bacteria in your gut, right? Yeah,
it's one of those things that is a balance. Yes, providing you have a suitable protection, then they will do the turnover job that you would expect for normal metabolic processes to carry on. The problem is when it gets out of hand, so that you start to lose the balance, you lose the protection, you get interference with the epithelial cells wherever they happen to be, and then you get all sorts of pathological things as a result of that.
So I presume that's why, you know, high sugar diet is not very good. Is that would that impact?
That's part of it, yes,
Yeah.
certainly.
Right. Is there any supplementation of the microflora? Can they get through the stomach? And will it settle on the intestine?
The thing that you have to look at is the natural microflora at different locations, because depending on, for example, in the stomach, as you would expect, because the pH is so acidic, the number of bacterial species is small. But as soon as you get to other areas, the mouth has a lot, as I think Jacob was saying. There are certainly in the small and large intestine areas where particular bacterial flora are common. So it's an adaptation. It depends where you are, and then you need to look and see which species are actually able to survive and what they actually do. Again, the function is important as well.
Great, thanks, Tony. Are there any more questions from anyone? If not, if we can just all say thank you to Tony for his time and for giving us today's talk.
You're very welcome.
Thanks, Tony.
And this is the last talk before the Easter. We have Easter coming this weekend. So we'll reconvene again after the break. So we're going to come up with a new list. Tony, I already have you on a date you've given me.
Yeah, if we could push that back a little, because
yeah,
I'm busy, very busy over Easter. So, but that's fine.